Okay, this next unit is about chemical reactions and energy and enzymes that help speed up chemical reactions. So we're going to start out with talking about chemical reactions. So this is in our textbook, chapter 6, and we're going to start out with this word metabolism. Metabolism, I'm sure you've heard of that, but basically what it is the sum of all your chemical reactions, in our body. And so every single reaction in your body is collectively your metabolism. So then let's look at two types of chemical reactions that we need to be familiar with. So two types of chemical reactions. So the first kind, number 1, this is called a catabolic. Or there's two different words to describe it, or exergonic reaction. Now, what in the world is that? So these release energy. Release energy by breaking down molecules into smaller molecules. So let me give you an example of in a living thing of this happening. So example is digestion in our digestive system. So example I'm going to use is protein. Protein, now test yourself. Can you think of what protein is broken down into? Pause it if you need to. Protein is broken down into amino acids. Because proteins are chains of amino acids. All right. So let me give you some characteristics about a catabolic or exergonic reaction. We're going to draw a graph here. Okay, so we have the axis here. And this is, we're going to put energy, and I'm not going to put numbers on here, just as you go. It's going to go up as you go over, and then this is going to be time. So from left to right is the passage of time. So in a reaction, you have the stuff that goes into your reaction and the stuff that comes out of your reaction. What goes into your reaction is called your reactants. So those molecules that are your reactants have a certain amount of energy. So I'm going to go ahead and I'm going to make this a bar graph here. All right. So these are your reactants. And then what is true at the end of the reaction is you end up with different molecules. Those are called your products. And so for an exergonic reaction, these products have less energy. So, the laws of thermodynamics say energy can be neither created nor destroyed. And so if these reactants have more energy than the products, that energy just doesn't disappear. It has to go somewhere. All right, so where does that energy go? I'm going to switch colors here. So that difference in how much energy the reactants had compared to the products, that difference there, that energy goes somewhere. Some is released as heat. Released as heat. Now heat is this random movement of particles, right? Heat in a living organism is not usable energy. It's randomness. It represents randomness. So when this kind of reaction happens and some of the energy is released as heat, I'm going to put in parentheses that this is unusable. But then some of that energy, let's do here, some of that energy is used to do work for the cell. So it's positive. They call this type of energy that's used to do work for the cell, they call it free energy. This is the energy available to do work, all right? To do work, I'm going to underline that. So all of this heat energy is not available to do work, all right? It's unusable. All right, so let me give you an example of a reaction that this is the case. Cellular respiration is an example where you have your reactants like glucose, where we break down glucose to get energy. So we have glucose. Glucose has way more energy in it than when we break down glucose. Remember that was C6H12O6. When we break it down, it gets broken down into water and carbon dioxide. These two molecules, while we need water to survive, we cannot use water for energy. There's not energy available in the water. So, same thing with carbon dioxide. So these guys too have less energy than our glucose. Now let's go back to this free energy. So what is true about an exergonic reaction, all right, is that it releases energy, right? And that some of that energy is free energy. That free energy, this word here, is represented by the letter G. So in an exergonic reaction, the delta G, whenever you see this delta, that means the change in, all right? So the delta is the free energy in the final products, so that's G sub F, minus the free energy in the initial reactants, that's G sub I. So that's your change. So can we see here, so this right here, this is GF, and this is going to be GI. And so when we figure this out for an exergonic reaction, if we just think of this as a math problem here, you see that the products have less energy than your reactants. So it doesn't matter how much energy is in here. This final number, all right, is going to be a lower number. So I'm just going to down number, meaning this is the lower number, minus the reactants have higher free energy, so minus a higher number. So therefore, mathematically, if you take a low number minus a higher number, what kind of number are you going to get every time? A negative number, all right? And so whenever you have a negative delta G, this means that energy is released. All right, so that's one type of reaction in which energy is released. Number two, all right, so number one was catabolic or exergonic. Now we're going to do number two here. Is called an anabolic reaction or endergonic. All right, so we're going to put this uses or consumes energy to build molecules. So just like before, I'm going to give you an example. All right, so this is, I'm going to use the proteins again. Building proteins from amino acids. So you eat a cow protein, you digest it into amino acids. The amino acids go through your bloodstream, go to your cells, go into your cells, and then you use it to build proteins. And so what does this look like in a graph form? All right, so we're going to do the same kind of thing that we just did. The x's are going to be the same. We have energy and time. And this kind of reaction, though, you still have your reactants, but your reactants have less energy. So these are your reactants than your products. Okay, so now if we look in, let me get my red here. Now we have an energy difference here where there's more energy in your products than you originally started out with in your reactants. And so where does the energy come from? Energy can be neither created nor destroyed, so it doesn't just magically come. So you have to have some kind of energy input, all right? So if we do the same kind of thing with our reaction with delta G, G final minus G initial. So this is the amount of energy final, this is the initial. So in this case here, your final has a higher number than your initial. So mathematically, you do this math equation, big number minus small number, you end up with a positive number every time. So if you have a positive delta G, what does that mean? It means the energy is consumed, meaning that you have an input of energy. So I'd like you to now, let's take a look at this handout here, all right? And it's going to illustrate how these two types of reactions kind of work together in our body, all right? So this right here is supposed to represent a cell, all right? So you've got, well, let me see here. So this is your cell membrane right here, right? Now, let me see here. Let me pick out my blue here. So let's say, so this is the cell membrane of a cell, just a cell in your body. And let's say, let's give a scenario here. Let's say we eat some starch. Starch is a chain of glucose. One glucose hooked to another glucose, hooked to another glucose, etc. Right? So lots and lots of glucoses. The first thing your body is going to do is break that down. So we're going to digest the starch. Now, how are we going to digest it? We have enzymes to help with that. And we're going to, those enzymes are going to help break these bonds between the glucoses. How do we break them? All right, we're going to add water. So when we add water, can you remember what that is called? Where you put an H onto one and an OH on another. If you're saying hydrolysis, you would be correct. The split using water. So at each bond here, a water molecule would go in to help to digest it. So now when we digest starch, this is what you end up with. Instead of one long starch molecule, you have many smaller glucoses not hooked together. So here's some 3 glucoses floating around here. So let's say that one of these little guys is a glucose. So all of these little guys are glucoses. high concentration of glucose outside the cell. So how is that glucose going to get through the cell membrane? Well, the cell membrane, remember, has a lipid bilayer and proteins. My next question to you is, how in the world does glucose get into the cell? Does it go through a protein or does it go through a lipid bilayer? If you're saying it goes through a protein, you would be correct. So right here, I'm going to put in here, they don't have this in the drawing here, but I'm going to put here that this is a transport protein. So it's going to allow it to go through. And that glucose from high to low concentration is going to go into the cell. Now, the thing is that there's two different things that you can use with it, two different ways that you can use this glucose. You can go this way or we can go this way. All right, so let's look at the first top way here. Let me get a different color here and we'll go back to red. All right, so this glucose can, now it's a small molecule, can be hooked together, all right, to make starch, all right? So this is starch. Starch, this would be, if it's in plants, in plants. What's the long chain of glucoses that we store in humans? Starts with a G. We hook a bunch of glucoses and we store it as glycogen. This is in animals. Next question, do you remember where we store glycogen? If you're thinking liver and muscles, you would be correct, all right? So this picture is showing you building other things. I use the example of eating starch. So starch breaks down into glucose. Glucose gets into your cells, and we can build to make large molecules like starch or glycogen. If we were eating protein, we digest the protein into amino acids. The amino acids would come in and then be put together to make human protein. So you eat a cow protein and we use it to all those amino acids to make a human protein. Or we could eat fat and we break it down into a glycerol and 3 fatty acids. Those enter into the cell and can be made, we can make a triglyceride. All right, we can do that with DNA as well as phospholipids. So everything that we eat can be broken down so that it can get, so it's small enough to get into the cells, and then we use it to make bigger molecules. So that's what this is. So is this an anabolic or catabolic reaction where we're taking glucose and building a larger molecule? We go back over here, building larger molecules, that's an anabolic, all right? Building is anabolic, so it's right here. This is an anabolic. Another word for anabolic, could you say it? Is endergonic reaction, all right? And so that is an anabolic pathway. Now the thing is that anabolic pathways need an input of energy. So where in the world does that energy come from to build it? And so that energy comes from a catabolic reaction. So I'm going to switch colors here. So I'm going to use green here. So this glucose can be used, you can store it, or you can use it for energy right away. All right, and so this is glucose. So this, what we do is we break down glucose. I'm going to write that here. Whoops, my pen is not, it's all dried up. I got to throw that one away. Let's try this one. Right here that it breaks down glucose, just C6H12O6 into, so we have some molecules here. These molecules are going to be, they're going to represent, in my example here, water and carbon dioxide. So this is the example here. When I said breaking down glucose into water and carbon dioxide, this is cellular respiration. It releases energy, all right? And so this is the energy. This is heat energy. This is the unusable kind. All right, so we got this little firecracker. All right, and so heat energy is unusable. But remember that some of the energy released is actually free energy and is available to do work. And so that's what this arrow right here is representing. All right, so this arrow is showing you that this is energy to do work for the cell. And so you can see that the catabolic pathway, breaking it down, provides energy for the anabolic pathway. And so this is called coupling or a coupled reaction. A couple is two. So one reaction drives another reaction. All right, so you have to have this reaction to provide the energy for this reaction. That's called energy coupling. All right, so now let's go ahead and go back to our notes here. I'm at the end of my page here, so I'm going to go to a different page, a new page. And if we recall, let me go back over here, we were on A, the two types of chemical reactions. So I'm going to start with B at the top of my new page here. So B, I want to talk about the energy coupling thing. All right, so exergonic reactions can supply the energy to drive endergonic reactions. So I'm going to give you a couple of examples, and I'm not going to do real molecules because I just want you to see what happens. All right, so number one, first reaction, we're going to have molecule A, whatever that is, plus molecule B. Those are your reactants. Then we have our reaction, and so now you're going to have your products, which are different. In this case here, A is going to hook to B. That's your end product, all right? So this, we're building a molecule. So this is an endergonic reaction. Now I'm going to ask you a series of questions here when we go over these, and I want you to play along. Don't try to just be getting through this to get your notes done. This is a learning opportunity. So this is an endergonic reaction. So my next question to you is, if we did a little graph, could you draw the bars to show the relationship between energy in reactants versus products? So if you need to pause that for a second, do that. In an endergonic reaction, the reactants have less free energy than your products. So if this is the energy and this is the time, that's what it would look like. My next question to you is, what would the delta G be? Is that a positive or a negative number? It's final minus initial, big number minus small number. So you are going to get a positive number every time. I'm going to quantify this and say that this reaction takes 3.4, and kilocalories is a unit of measurement for calories, all right? So, or for energy, sorry. So kilocalories, 1,000 calories, and the calorie is telling you how much energy is in the being released, and this is per mole of the substance, all right? So if you have a mole of the reactants reacting, this is how much energy is released. All right, so that's your endergonic reaction. Then let's look at an exergonic reaction. Exergonic reaction is using ATP. ATP stands for adenosine triphosphate. It has an adenosine molecule, I'm just going to put A, with triphosphate means 3 phosphate groups. And we'll look at this a little bit more later. But how we get energy is we break it down, we break the phosphate off. So plus water, because remember, to break a bond, you have the addition of water. I can do that here. So here's water. You go on H2O, and this bond breaks right here, right? And so what you end up with is you end up with ADP, which is adenosine diphosphate with only 2 phosphates. And then a P sub I, that just means the I means inorganic phosphate. It means that it's just not attached to a molecule. It's floating around here. So this is an example of an exergonic reaction. So it's going to release energy. And so if I do that little graph, could you do the bars? Reactants versus products. The reactants have more free energy than your products. That's how you get energy that's released. So what's our delta G going to be here? Is it going to be a positive or a negative number? Final minus initial, you're going to get a negative number. And I'm going to quantify this one as well. Let's say that this is actually a real number. Breaking down ATP is 7.3 kilocalories per mole. So relating this back to the energy coupling that I was talking about here, that a catabolic reaction that releases energy drives an anabolic reaction. So ATP, the breaking down of ATP, releases 7.3 kilocalories. So that's the amount of energy, right? The negative just means that it's releasing. So this is a number, a quantity of energy. This reaction only needs 3.4 kilocalories. So do you see that this one is releasing more than enough energy to provide the energy to make this one happen? So therefore we can think of this as, if we say this happens with this and this is going to drive this reaction, if you add these two together, if we think of it like a math problem, all right, negative 7.3 plus 3.4, you're going to get a delta G of 3.9 kilocalories per mole. All right, what this means, all right, I'm going to write here that this is a coupled reaction, 2 reactions working together. Coupled reaction. Whoops, I'm sorry, I'm a little off. New word there. All right, the overall delta G is negative. So together the reactions are exergonic, or I'm going to introduce this to you, spontaneous. We consider reactions that are exergonic, we call them spontaneous reactions. So going back, let's go back to our first page right here where we talked about catabolic or exergonic reactions. I'm going to put a little star here that these are called spontaneous reactions. Okay, and drive the endergonic or catabolic reactions. Now the thing is that most of the time in the cells, a reaction, would happen too slow for for enough of it to happen to keep the cell alive. So in comes the help of enzymes. So this is going to be letter C over here. So I'm going to introduce enzymes to you. Enzymes, do you remember if they're carbohydrates, lipids, nucleic acids, or proteins? They are proteins. Can speed up. I didn't leave a lot of room here, so I'm going to come over here. Speed up both types of chemical reactions. Both endergonic and exergonic. So I'm going to put a little 1 here. And write some things about enzymes. Enzymes are biological catalysts. A catalyst, I'm put in parentheses, is anything that speeds up a chemical reaction. That's what a catalyst is. Let me give you some examples. Examples include heat. Heat can speed up chemical reactions. Light can speed up chemical reactions. And enzymes. So the enzyme is a biological catalyst. So enzymes are catalysts, but not all catalysts are enzymes because it can be other things like heat and light. All right. So enzymes are one type of catalyst that can speed up chemical reactions. Let's put a number 2. Another thing is that enzymes are specific. For a certain substrate. New word. What's the definition of a substrate? Let's put in parentheses here. This is the reactant that an enzyme acts on. So let me give you an example of a reaction. So we're going to look at a reaction where we're going to take sucrose, that's table sugar, plus water, H2O, and we're going to break it down into glucose and fructose. So the table sugar can, if given water, you can break it down into these two things. Now, sucrose, I'm going to do red for that. Sucrose is os means it's a sugar. If you recall, it's a disaccharide. So I'm going to go ahead and draw like this and put a G and an F. It's glucose hooked to another to a fructose. So it's a disaccharide. So this right here is your substrate. Now what's going to happen is an enzyme is going to speed this reaction up. All right, so I'm going to do another color here, and I'm going to do, I have just enough room to make a little drawing down here. I'm going to draw some things down here. So you have right here, I'm going to do an enzyme that has a specific shape. Notice the shape is similar to the substrate where it can fit in. So this right here let's label this. This is the enzyme. This enzyme that speeds up this particular reaction. Remember that they're specific. So an enzyme that speeds up this reaction is going to be different than an enzyme that speeds up another reaction. So this enzyme is called sucrase. ACE, whenever you see the ACE at the end, that usually means that it's an enzyme. And they try and make the name sound like what it's helping to speed up. So sucrase sounds like sucrose. So the sucrase enzyme helps to break down sucrose. Now, if I go back to my sucrose, this has a similar shape as right here. All right, and this can go ahead and fit into this. This part of the enzyme right here is called the active site. This is where the substrate fits in. All right, now to relate this back to macromolecules, that enzymes are proteins, right? So I drew this as a boxy little shape, but realistically, remember that the protein would be a chain of amino acids, so there'd be interactions between the R groups, and there'd be beta pleated sheets, and so on and so forth. All right, so realistically, this is a chain of amino acids, is what that protein is. All right, just to relate everything together here. All right, so then, draw another picture here. So once this binds into this, all right, so we're going to look at, let me see where I can draw. I'll draw it right here. All right, here's my enzyme again. Now the substrate is in there, right? So here's in the active site, glucose and fructose. They call this the enzyme substrate complex. And then what's going to happen is this enzyme is going to help break down that glucose, or that sucrose, sorry, into glucose and fructose. So here's your enzyme, active site still intact here, but now what comes out is different molecules. So I'm going to switch, I'll switch colors here. I'll do pink here. All right, so this is going to come out. And you're going to have glucose by itself, and you're going to have fructose by itself, no longer hooked together. And these are your products. All right, so that is this right here, glucose and fructose. So your end products right here. All right, and so what I want to point out, though, although my picture doesn't show it because I'm not a great artist, but is that what I wanted to show you is that the enzyme in the active site is still intact. So this enzyme can be reused, and if there's another sucrose can bind to the active site, and the same enzyme can break down lots and lots of sucrose molecules. So I'm going to write here that enzyme remains unchanged and reusable. Unchanged and reusable. So because it's not consumed, it's not actually a reactant and can be reused, but it helps to speed up this chemical reaction. We usually write the name of the enzyme up here, sucrase. So anything that is on or below an arrow is not used up in the reaction. It helps to speed it up, but it is not a reactant or a product. And so this can speed up this reaction, and the next sucrose can speed up that reaction, the next sucrose, and so on and so forth. It goes over and over and over again. All right. And so this is where I'll stop in the video, and we'll look at some more things about enzymes later.